Good evening and welcome everyone. Today is our 1684th day of continuous webinar. Welcome all the participants and the faculties. Today's our guest is Dr. Suraj Singh Bhadoria. He has already presented his research papers in IFPH many times, and he is MD and PhD scholar, assistant professor in the Department of Homeopathic Pharmacy in Parul Institute of Homeopathy and Research, Vadodara, Gujarat. He made lot of presentation in various forums. Today's topic is advancement in homeopathy. So I am welcoming Dr. Suraj Singh Bhadoria. You are the co-host now. You can.
Yes, ma'am.
You're welcome. Please.
First of all, good evening to everyone. Today we are going to discuss about advancement in the homeopathic pharmacy in the field of homeopathy. Ma'am, can you share the PPT, ma'am?
Yeah. I'll share.
Yes. Now, today this topic plays a major role. What is the advancement? Through this research work, we are going to have the basic progressive development in the homeopathic pharmacy in terms of nanoparticles, because nowadays this nanotechnology plays a major role for the development, for the progression, for the technology, for making our pathy as a evidence-based medicine. So the project of this work is preparation of nanoparticles from toxic human blood serum with the help of homeopathic medicine, which is Thuja occidentalis mother tincture, which is prepared by the plant leaves of that Thuja after the pulverization or the potentization. So this is our main objective. Now moving towards the abstract part, we prepare the nanoparticles from the toxic human blood by the Thuja occidentalis mother tincture as a 1x dilution in the process of potentization, and the analysis or the standardization were completed by the SEM analysis. SEM is basically a type of electron microscope which stands for the scanning electron microscope, and the compositional determination of this nanoparticle were done by EDS scanning in the part of electron microscope. Moving towards next part. Now basically this human blood, it plays a major role in our body in terms of the various compositions, in terms of various composition of elements, in terms of various composition of cells, the cellular structure, the tissues. So these all are the subunits of our living entity. So what we are going to do, we are going to synthesize the nanoparticles from this proteinaceous structures, from this cellular structures, from this elementary details which are present in our serum blood, in human blood. And after preparing these nanoparticles, now they are going to act as an application of drug, and then it enters into the drug delivery system. So these are our major aspects of this research work, why we prepare the nanoparticles from human blood serum. Next slide. In methodology, now this is a type of nanotechnology which plays a major role in our homeopathy, because still homeopathy is a pseudoscience. It is a alternative medicine. It is not said to be a evidence-based medicine. That's why we prepare the nanoparticles in our ultra high dilutions, in our various different mother tinctures. So the site of study is Parul Institute of Homeopathy and Research, Homeopathic Pharmacy Laboratory, and Micro Nano Lab of Parul University. In Parul University, we have this facility, we have this Micro Nano Lab where all the researchers are going to have this facilitation for the nano screening, compositional determination by the SAM analysis and the TAM analysis. The duration of this study is around 15 days. And the sample which are used for this type of research work is human blood serum, and the medicine which is used for this particular nanotechnology is Thuja occidentalis Q. So here it is a green synthesis. Generally in the nanotechnology, in the nanoparticles, there are certain procedures there are certain methods through which we prepare the nanoparticles. So here we use Thuja occidentalis Q because it is generally prepared by the leaves of that particular plant. So they are showing a green synthesis. So this is the first time when we prepare these nanoparticles by the green evolution. So the tool which is used for this particular research work is electron microscope and the centrifuge machine for separating the blood serum and the electric potentizer machine. Analysis were completed by SEM analysis. The basic criteria for this analysis is to identify the micro and minute structures which are present in our green synthesis in our human blood serum, and also determine their elementary details like the carbon percentage, like the oxygen percentage, sodium percentage, and some other elements which are present in our human blood serum. So these are the major components of this research work. Next slide. Now this is a SEM. Basic this analysis of this instrument, it is used to determine the various microstructures. It is used for the material research in terms of nanotechnology, in ceramics, in compositions, in polymeric materials, in geology, in dental materials, biological substances, and the soft materials. So these are the basic application of this SEM. So it is very, very important and very sensitive instrument. This identify the minute and the microstructure in terms of a various wavelength of microns in our given solvent or the media. Next slide. Now this is a sample where which we collect the human blood, and after collecting this human blood from the healthy individual, healthy volunteer, we separate this under the centrifuge machine. So after centrifugation, we have three main chambers. First is for serum, second is for buffer, and third is for the basic RBC chamber. So in serum, we have all the protein part and electrolytes. And in the buffer part, they have all leukocytes and WBCs. And in the lower segment, lower chamber, we have RBCs only. So after collecting this serum, now it is going to mix with the Thuja occidentalis mother tincture, which is taken as around 5 ml. Afterward, we mix this Thuja occidentalis mother tincture with the human blood serum. So as we know, while preparing the mother tincture, the main solvent system is hydroalcoholic base from maceration or the percolation. So generally we prepare Thuja occidentalis by the process of maceration because it is gummy, it is juicy substance. So we use maceration. So after preparing this Thuja occidentalis Q by the maceration, they have hydroalcoholic base, and we know when this alcohol mixes with the blood or the serum, so it becomes a toxic, just like a patient of cirrhosis or the alcoholism. When there is a raised level of alcohol in the blood, so that may cause the toxicity in the blood serum. So this is a basic composition of this Thuja occidentalis and serum blood which is collected from the healthy volunteer. Next slide. Now after mixing this Thuja and the blood serum, we go for the process of digestion by hot water bath. So why we go for indirect heating? Because generally this indirect heating, it preserves all the alkaloidal property and the basic composition and the elementary details which are present in our given solvent system. That's why we go for the indirect heating. But when we go for direct heating, so they destroy the elementary details and the alkaloids and the compositions which are present in our solvent or the media. So we use hot water bath for indirect heating for at least 10 to 15 minutes. Afterward, we go for the potentization. After potentization, we make this 1x potency. Moving towards next slide. Now, after this 1x potency by 100 downward strokes with the help of electric potentizer machine. Now, this is a SAM analysis. Now, you can see these are a different crystalline arrangement of the nanoparticles which are prepared by the green synthesis with the help of Thuja occidentalis Q, which is homeopathic mother tincture, in human blood serum. So you can see this is just like a small clustering growth which are forming a heterogeneity in the biomolecular system. So this heterogeneity in the biomolecular system shown the pattern of arrangement in the nanoparticles. Next slide. Now this is images in 5 micron and 1 micron. In 5 micron you can see a heterogeneous clustering growth, whereas in 1 micron you can see a small lobular and a small tubular shape which is around 1 micron in that particular heterogeneous or cluster pattern of human blood serum, which is prepared by Thuja occidentalis Q. So these are all the images which are analyzed after the SEM. Next slide. Now afterward, this is again a SEM analysis in 2 micron and 15 micron. So these are in a different range, in a different microns. When we prepare the nanoparticles, so we are going to analyze this structure in a different range so that we can analyze and we can make a different pattern of arrangement. We can analyze this nanoparticle in a different shape, in a different structures, in a different chains, in a different cubic shape, so that we can know about their morphological features. Ultimately, in electron microscope, these electrons, when they are going to hit on the morphological surfaces of the particles, so they will give you a certain dimension about their morphology, about their structure. So this is very, very important part. When we do nanotechnology, when we talk about nanotechnology, so it should be in all aspect. Next slide. Now it is a EDS. Generally, this EDS, it reflects the compositional determination of that particular nanoparticle. What are the compositions which are present in that particular nanoparticle? So this is the image for the EDS analysis of toxic human blood serum, which is prepared by Thuja occidentalis Q. Next slide. Now these are the elementary detail. Now you can see the level of potassium, the level of oxygen, the level of chloride, sodium, potassium. So in the molecular weight, in the atomic percentage, you can determine this compositional value of these different elements in our nanoparticle, which is prepared by toxic human blood serum under the process of potentization or dynamization. Next slide. Now you can see carbon in EDS scanning, the atomic percentage is 67.59. Oxygen in EDS, the atomic percentage is around 29. Sodium in EDS, the atomic percentage is around 1.46. In chloride, the EDS scanning is around 1.5. In potassium, the EDS scanning is around 0.45. So it is already a distribution of various composition and the elementary detail. So what it indicates? It indicates that whatever we prepared, it should have all these elements, because ultimately our body is made by all these carbon atoms, oxygen, sodium, chloride, potassium. So they are having a different composition in a different atomic number, in a different molecular weight. So when this particle possesses all this elementary composition and the molecular weight of such elements, so they are going to trigger and progress in our cellular events. If they have lack of oxygen, carbon, sodium, chloride, potassium, so ultimately the efficacy of that particular nanoparticle is going to decrease. So we have to understand this particular composition also. When whatever we prepared by any medicine or any source or any methodology, it should have all this proper elementary composition. When they are in a equilibrium and when they are in a different percentage, in a different atomic percentage, in a different molecular weight, molecular percentage, so they are going to show their some significance and efficacy towards a particular cellular event. So this is again a very, very important part for nanoparticles. Next slide. Now this is again a elementary detail of nanoparticle in a 100 micron. In 100 micron, you can see the carbon EDS scanning is around 58.83. The molecular sigma weight is around 1.4. Whereas in oxygen, the EDS scanning is around 33.62. That is the molecular weight of that particular nano and element of oxygen. And the sigma molecular weight is around 1.19. In sodium, in EDS scanning, the overall molecular weight percentage is 2.43. Whereas the weight of the sigma elementary detail is 0.15. In chloride, the EDS scanning is around 3.84 in the molecular weight percentage, whereas the sigma molecular weight is around 0.18. Whereas this potassium in EDS scanning, the molecular weight percentage is around 1.28 and the weight of the sigma percentage is around 0.11. So overall, this molecular composition in a different sigma weight percentage of different elementary details were determined by the EDS scanning. Next slide. Now this is a structure of nanoparticle of toxic human blood serum from Thuja occidentalis in 100 μm. Next slide. Now this is a compositional detail. Now you can see there are differences in 50 μm, in 100 μm. Now you can see the basic elements are carbon and oxygen, whereas in previous slide they are more towards potassium, chloride, sodium. But in this particular range, you can only have the carbon and oxygen. Next slide. Now the molecular weight is around in carbon. The EDS scanning, it shows the atomic percentage is around 65.88, whereas in oxygen the EDS scanning is around 34.12. So overall, these are the atomic percentage, atomic weight, and the molecular weight of this carbon and oxygen in this particular range of nanoparticle of toxic human blood serum. Next slide. In another aspect, you can see the sigma weight percentage of this carbon. So the molecular weight is 59.18 in EDS scanning, whereas in weight of the sigma of that particular carbon element is around 0.51. Whereas in oxygen, the EDS scanning, the weight percentage is around 40.82, and the weight of the sigma molecular weight of oxygen is around 0.51. So it is in the distribution of 100%. So you can see here the carbon element, it is frequently seen more as compared to the other elements. So what it indicates? It indicates that that particular nanoparticle shown their efficacy more towards the carbonitrogenous constitution, because ultimately in our organon of medicine we have a different types of constitution, or a diathesis or a miasm. So what it indicates, generally when we have carbonitrogenous, oxygenoid, hydrogenoid constitution, so generally we have a different elementary detail in a different molecular weight, in a different atomic percentage. So when we prepare this particle and when they have more carbon, so they are showing more efficacy towards carbonitrogenous constitution. When they have more molecular weight towards oxygen, so they are more having efficacy or their pharmacological action towards the oxygenoid constitution. When they are in more towards the H2NO water molecule, so it's said to be a water memory. So it is more towards the hydrogenoid constitution. So these are the basic what we can say correlation of this molecular composition of a different elements in respect to the different types of constitution under nanotechnology. So again, it is very, very important, because when we talk about PG, so PG, now there is new CBTC curriculum. So suppose if there is a chapter of miasm, so how we will explain the scientificity and the evidence of miasm? So generally miasm, they are just like a mRNA. So in the roundworm, there is mRNA, and that segment of mRNA is going to transmit it from one species to another species, one generation to another generation, and that segment of mRNA carry the memory from one species to another species, and this will act as a miasm. So this is a what we can say the approach of homeopathy towards the scientific base, towards the evidence-based medicine. So we have to nourish our PG students, not only PG students, the UG students, the PhD students, towards such type of practice, so that we can make our pathy as a evidence-based medicine. So similarly, nanoparticles also, when we talk about the types of constitution, so it depends on the basic compositional value of this particular nano. So when they have more carbon, so they are more towards carbonitrogen. Moving towards next slide. Now the basic conclusion of this research work is, this work is very novel and very new for us, because we don't know that when we mixes and have the Thuja occidentalis mother tincture with the human blood serum, so what are the heterogeneity, what are the molecular composition, what are the elementary composition, what are the basic pattern of these nanoparticles. So this is a again very important work, and it reflects more evidence in our homeopathy, because ultimately homeopathy is just like people thought that they are a placebo effect, they are a magic effect, but no, in ultra high dilution also you can have some concentration with the help of UV spectrophotometer, with the help of Raman spectroscopy, with the help of various instruments which are generally used for the standardization. Similarly, when we synthesize this nano, when we synthesize this micro and the minute structure, so they will give you certain memory, and they will reflect a certain elementary composition. So this will reflect their efficacy also, and this will also reflect their pharmacological action also, because when we talk about nano, so they have a very specific action, whereas in a cluster molecule they have no specific action, it is going to act on the whole body. So this is a basic difference. When we talk about cluster, so they have action on top to bottom. When we talk about nanoparticle, so they act on a specific viscera, on a specific cell, on a specific tissue, on a specific epithelial arrangement. So this is a basic difference. Why we are shifting towards nano? Because nano will give you a specificity. It will give you a specific order in terms of their pharmacological action, in terms of their chemical nature, in terms of their physical property. So this will very important and fruitful result for making our homeopathy as a evidence-based medicine. So this is again very important practice, and we are again very thankful to the Parul University, trustee of Parul University, Dr. Komal Patel Madam, Dr. Parul Patel Madam, Dean Dr. Purav Desai sir, for concluding this research work and for supporting us and for providing all these research platforms for synthesizing, for analyzing, for quantification, for qualification, and not only completing this, also publishing it in a reputed index journal. Next slide. Now, again, we are thankful to the faculty of Micro Nano team of Parul University. Next. These are our references, and this is already published in an international journal. And again, we are very thankful to Parul University for this platform, for providing this research work, and for completing this research work and publish it. Next slide. Now, this is our publication detail. It is published in this volume 7, issue 3rd, part A in 2025. The title is Preparation of Nanoparticles from Toxic Human Blood Serum by Thuja occidentalis Q after potentization. And the main support and the main pillar of this research work is Rathwa Rameshan. He is our technician, lab technician, who work in our hospital in IPD section of Parul Institute of Homeopathy and Research. And this is again a very important to know about the what we can say the beauty of homeopathy in respect of the allied sciences. So whenever we work with some other pharmacy or the agro homeopathy or the veterinary sciences, so we always learn more and more from other faculties also. So again, I am very thankful to our lab technician Rathwa Ramesh Bhai, and this work is published in the International Journal of Chemical Research and Development in page number 42 to 50, and it is again a very important part for not only for synthesizing it, for also publishing it. Our next goal is to show the efficacy of this nanoparticle in in vitro study and in vivo study, animal model, mice, for showcasing their efficacy. So again, it will be a very challenging job for us to just have a specific action of this nanoparticle, which is prepared by human blood serum, toxic human blood serum. So again, we are very thankful for this work, and we are also thankful for IFPH platform for showcasing, for explore our knowledge, explore our experience, our research work in the homeopathic fraternity, so that we can all make a what we can say, a group or a chain moving towards the evidence-based medicine. So this is again very important for us, and we are again glad to share this work with all the IFPH members. And afterward, we are again in a process for the evidence-based medicine by various tools and the models in animal, in vitro study. So again, this is very important for us. Our next part, we are again in a preparing a homeopathic culture media. So again, it is very important for us. Nowadays, we have no such culture media in our homeopathy. Generally, we use culture media like agar agar, blood agar, chocolate agar. But in homeopathy, this is the first time when we prepare a homeopathic culture media by the homeopathic medicine to promote the growth of some other bacilli or the fungal variety like E. coli, S. aureus, streptococci, and Candida albicans. So again, we are in the process for preparing the homeopathic culture media. So again, we are thankful for this research work, for this publication, and for all the members of IFPH.
Thank you very much. Actually, many of us have no idea about this research work and all. It's a you have shared a wonderful thing here. At least we get some idea about that, and we can show to the people, those who are saying homeopathy is not evidence based. Now we have in IFPS forum we have research scholars in our forum. I'm so proud of you, Dr. Suraj Singh. Now, Dr. Mahmood Saidun raised his hand. Sir, you can ask.
Audible, please.
Yes.
Yeah, thank you very much.
Yes, yes,
It's
sir.
very interesting. Thank you very much for the introduction of amazing research. But how can we guarantee the toxicity or the safety on human cells of the nanoparticles? It is already known due to the high energy invested in preparation of the nanoparticles. It has a lot of energy, and this energy is sometimes toxic for the human body or human cells. Can we guarantee the safety of the same on human cells?
Sir, basically, after preparing such a nanostructures, our first study is phase 0. In phase 0, we are going to study this action of nanoparticles in vitro trial in particular cell line or in animal model like rats, rabbits, mice. So when we talk about toxicity, so first we have the phase 0. When there is no toxicity in phase 0, then we are going to enter in phase 1, that is clinical trial. So again, it is under the process, sir. We are just synthesizing this nanostructures in the green preparation, green synthesis by the toxic human blood serum by Puja occidentalis Q. Afterward, we enter into the in vitro or in vivo trial. And after completing this, definitely we will share the toxicity analysis of this nano also. And if there is no toxicity, then afterward we will enter into the human trial also.
Yeah. I have another question, please. Can I?
Yes,
Yes, sir.
sure.
Yes, sir. Definitely.
Yeah, thank you. Can we use your homeopathic media agar for cultivation of organisms we are used to do in microbiology?
Yes, sir, definitely, sir. We are again in that particular chamber also, sir. We have one medicine, sir, Echinacea mother tincture. Generally, this Echinacea we use for this antiseptic, antibiotic, antifungal in cases of eczema and some other skin lesion. But what we found, sir, Dr. Henneman, sir, already told that every molecular system, when we talk about biomolecular system, so every molecule has a biphasic nature. So in first phase they are promoting, and in second phase they are also inhibiting that particular bacteria, that we can say a law of similars. So similarly, when we prepare this homeopathic medicine in a such a manner, so that they can not only inhibit the growth, they can also promote and flourish the growth of all the microbes and the fungal variety. So it is already told by Dr. Henneman in various edition of Organ of Medicine. And recently, sir, it is very shocking results, sir. We had this what we can in this echinacea culture media, homeopathic culture media in E. coli, in S. aureus, in Streptococcus, and in Candida albicans, which is a fungal variety after 48 hours. So again, this is a very shocking for us. So after entering in this biomolecular system, in this chemistry, we know that every molecule behaves a biphasic nature. So in first phase they are promoting, and in second phase they are also inhibiting that particular base also. So yes, it is possible, sir, but there should be a proper compositional detail also, because for culture media we should have more carbon, we should have more oxygen, because there are certain aerobic bacteria. So we should have more nitrogen, more vitamins, because in some culture media the allopathic, the microbiologists, they use a certain minerals or the vitamins to enrich this particular culture media. So we can also use this as a supplementary, but our main medicine is our homeopathy. So this will be our one, what we can say, a novel work for this homeopathic culture media. Yes, sir, it is possible.
Sorry, sir, and we have a very important concern is to produce the antibiotics from the natural sources, from the homeopathy medications. It will break through globally if we can manage to isolate antimicrobials from the homeopathy drugs. It's not that important. It is very important, and it is a breakthrough. It will make a lot of industry on that.
Yes,
So
sir. Yes.
we should give it an importance.
Yes, sir. Because sir, nowadays people thought that homeopathy is just like a ultra high dilution. It is a magic. It is a placebo. But ultimately, sir, in more dynamic form, the biphasic, the character of this particular molecule is more enhances. So you can see the number of varieties of that single molecule in a different dimension. So when we prove this, so ultimately homeopathy is just like a evidence-based medicine. So we can prove that we can have this scientificity in our homeopathy. So again, it is very important for us, sir.
Yeah, thank you, sir. Thank you, sir. Although I am not a homeopath, but I have interest. I am not also not an MD, but I have interest in homeopathy. Thank you very much. Shukran.
Thank you so much, sir. This word is very important for us, sir.
Thank you. Thank you, sir.
Thank you, sir. Thank you for your questions. And if anyone have questions, please raise your hands. Dr. Leda Manoharan, if you want to appreciate, you can directly unmute yourself and you can appreciate. Dr. Danesh, are you there?
Thank you, Dr. Ajini Malekar. Thank you, Dr. Suraj Singh Bhadoria. Very thankful to you, because our homeopathy system is facing a very challenging time nowadays, because all the world is needed evidence-based system of medicine. But we have the homeopathic medicine totally evidence-based, but it is in a nano level, it is working, but it is not proved, like you said, in the nanoparticles, and the way of medicine worked in different system of our body. And you are now Puja Occidentalis applied 1x for proving, and that is very appreciable thing. Thank you very much, doctor. You continue your research, more and more medicine for this type of presentation is also, we are very, very appreciable. And thank you very much.
Thank you so much, ma'am. Thank you so much. Your words are more valuable for us, ma'am, because nowadays, when we celebrate this 10th April for our Hanuman sir, for our father, so again, it is very painful for us that still homeopathy is not evidence-based medicine. So why it is not evidence? So in that manner, again, we are doing so many activities with our UG students, with our PG students, with our PhD students, with various faculties also, faculty of repertory department, different department in our university. So again, it is a huge, what we can say, a mountain. And after crossing this mountain, definitely we will have a very good charm and a diamond to make a homeopathy as a one of the best and the scientific evidence-based medicine. But it requires more and more hard work, because ultimately, ma'am, when we prepare this nanoparticles and this nanotechnology, so it is very challenging job for us, because in some time, in some preparation, there are more cluster and more bigger or in more homogeneous and shape and size of molecule, so there is no nano. So again we are re-preparing the nano, again we are re-standardizing it, again we are re-quantifying it and re-qualifying it. So it is a very huge and a big process. So again, it is just because of blessing from you all people, and we always appreciate the forum of this IFPH, in which we are just sharing our experiences in terms of clinical practice, in terms of this research work, in terms of the beauty of this homeopathy. So again, you people are also just like supported for us. Without you people, we are not completing this work. So again, the appreciation part and the support and the, what we can say, the pillar of this homeopathy is always IFPH is a great platform for this, ma'am.
Thank you very much, Doctor.
Dr. Mariam Salcedo, are you there? Dr. Ram Parvesh sir?
Thank you, Dr. Anjani, and congratulations, Dr. Suraj Singh Bhadoria.
Thank you
Suraj
so much,
Singh
sir. You are
Bhadoria is
always.
Suraj Singh Bhadoria is a dynamic person, and he
Yeah.
built the homeopathic
Dynamic,
medicine.
young, talented person.
Built the homeopathic medicine, nanoparticles. Very
Yeah, he
good
had done many
describe the
research and sharing all the researches in IFPH. And actually, the Parul University and the faculties of Parul University are supporting us very much and giving more knowledge to us. Actually, the purpose of IFPH is to share the knowledge together across the globe. So, Parul University is taking a good part to do that work successfully. Thank you so much.
Suraj Singh Bhadoria is
Yes,
a
sir.
great experience and great lectures, pharmacy department. Very good described. So very thanks, Suraj Singh Bhadoria, again presentation. Every presentation is a dynamic. Very thanks.
Thank you, sir. Thank you.
Thank you. And Dr. Dani, anyone else have questions? I think there is no questions, because actually for asking questions, we should have some idea about the researches and all.
So,
Yeah.
ma'am, can we discuss some basic thing how we prepare the nano, so that we can cover this part, ma'am?
Yes, sure.
Yes, ma'am. Ma'am, basically, when we prepare the nano, the first criteria is our source from which we prepare the nano. Either it is from plant kingdom, mineral, or metallic group, non-metallic, animal kingdom, sarcode, nosode. So the source plays a major role while preparing the nanoparticle. So if it is a metallic group like zincum, ferrum, so they are more potent for making these nanoparticles. So this is again important for us. First thing is the source where we prepare the nanoparticles. Second thing is the preparation part. how we prepare the nanoparticles. So there are different methods. First is like a physical method, second is chemical method, third one is the polymerization. So there are different methods through which we can prepare the nanoparticles. So in homeopathy, the biggest challenge is the process of pulverization and the potentization. We already have this method of dynamization. So the preparation of nanoparticle in our homeopathy is one of the biggest challenge, because in our ultra high dilution, when we talk about 1m and 10m, so they are already above the Avogadro number. They are more diluted. They have only energy part. So to bring some mass and the nano in this particular ultra high dilution is the biggest challenge. So when we talk about potency, again in the preparation part, the potency of that particular medicine plays a major role. If it is mother tincture, then it can be easily prepared, because we have pulverization, we have a electric potentizer machine, electric trituration machine. But when it is 1m and 10m potency, so to make the mass particle in that particular ultra high dilution is very challenging job. So the potency plays a major role. If it is mother tincture, so they can be easily created. And if it is ultra high dilution, so they are very, very difficult to create some mass. And the third important thing is media or the vehicle which are used in the process or the preparation of these nanoparticles. If it is distilled water, so they can be easily contaminated and they are not a good preserver. They can mix the basic ingredients or the compositional value of that particular drug substances, but they are not a good preserver which preserve the chemical and the physical property of that particular molecule. But this alcohol base, it is one of the good preserver. So always we have this alcohol for this preparation. Alcohol is one of the best preserver, and their stability is more better as compared to the distilled water. And if we talk about oil base, so again in oil base, the preparation of nano is again very, very difficult and challenging job, because in alcohol the viscosity is more, it is more dense as compared to alcohol and water. So in dense form preparation of nano, somewhere they are overlapped, somewhere they are hidden. So in oil base, again nanoparticles are very difficult. So these are the three different media. We have to know about this. What are the media while preparation of these nanoparticles? So if it is very less in the viscosity, if the viscosity is very less, so they are easily prepared. If the viscosity is more, so they are not easily prepared. So this is about the preparation part. Afterward, the stability and the time interval. Whenever we prepare any external application like lotion, ointment, liniment, hair oil, so it should has a certain stability. It should have certain what we can say stability in their chemical property and the physical property. It should not change their color, odor, texture, consistency. So similarly, nanoparticles also, the consistency and the color, viscosity, it is changed after preparation, but it should not like too frequently varied and change as per the temperature, pressure, and various external what we can say the stimuli. It should be like stable. If it is stable, then their efficacy would be like more and more effective. It is, if it is not stable, then efficacy is less. So stability part is the major criteria, and the next part is the
Doctor,
analysis part.
before
Yes,
moving to the next part, Dr. Mariam Salcedo from Bolivia, she has question.
yes, yes, ma'am.
Doctor, you can ask.
Thanks, Dr. Angie, and thanks, Dr. Suraj. It was an amazing session. I really enjoyed it and I learned a lot. And I have two questions, please. The first one is if can nanoparticles have different target organs? The first one. And the second question is it's just a thinking. Homeopathic remedy has different compounds. For example, Thuja occidentalis has different active ingredients. So the nanoparticles are made from all active ingredients or just for an active ingredients? What do you
Ma'am,
think about it?
yes, ma'am. Ma'am, for second question, the answer is basically when we talk about homeopathic preparation, so we use Thuja occidentalis as a bulk. Like we have both this primary metabolites and the secondary metabolite. The active principle of this Thuja occidentalis is Thujon, alpha Thujon and the beta Thujon. which are going to show their efficacy on the any cellular model or the living entity. So what we are going to show, we are preparing this nanoparticle as a bulk, because this is our laws and the principle while preparing the homeopathic medicine. When we use only thujon as an active principle, so this will not show the beauty of homeopathy, because while preparing mother tincture we use the whole pulp and the extract of that particular source, and while extracting it, it should be dissolved in a hydroalcoholic base for 1 week, for 2 weeks. Afterward we go for the filtration. So similarly, while preparing the nanoparticles in that particular media, so it should be in a bulk, in a composition. It is a holistic approach. When we took only thujon as an active principle, so it is just like a modern pharmacology. So there is no difference between homeopathy and modern pharmacology. So it should be separated, because when we chosen only one or a specific one, so it is not homeopathy, because our law is individualization. So we have to fix towards our principle, so that we can show efficacy more and more effective with the holistic approach rather than the specific one. And the first question is, ma'am, again repeat, you can repeat first question, ma'am. First
Thanks,
question,
Dr.
ma'am.
Ajini. Yeah, I understand. I totally agree with you. But my question is that in the nanoparticles, you found the whole tush, or are just tujon, alpha tujon, beta tujon as nanoparticles? This is the second question. And the first one was if can nanoparticles has different target organs?
Yes,
So
ma'am. Basically, ma'am, nanoparticle, when they are nano, so they have a specific organ. Because when we talk about clusters, so they are not in a specific part. Just like, for example, in nowadays, the modern pharmacology, now they are shifting towards individualization. So what are individualization? They are using nano technology in cancer treatment, in the meta-analysis, in the metaplasia. So they use a specific medicine for targeting only cancer cell rather than the other somatic cell. So now modern pharmacology also shifted towards a particular ground. So when the particle is nano, so definitely their action is more and more finest and specific and faster as compared to the cluster one. So similarly, when we prepare this nano from the homeopathic medicine, in mother tincture or in ultra high dilution, so definitely their action is more precise and specific one. Because ultimately, why the homeopathy is not evidence based medicine, it is pseudoscience, because we have no any specificity in the doses, in the posology, in the concentration of drug, in the action of drug. That's why homeopathy is just like a pseudoscience. They are not accepting homeopathy as a evidence based medicine. So what we are going to do, we are going to make our homeopathy as a specific one. So when it becomes specific, so they are going to act as a specific molecular system as compared to the holistic approach. So when it shown certain specificity in their therapeutic value, so that WHO and the whole world accept this homeopathy as a evidence-based medicine. We know that this is the holistic approach and the individualization. These are our beauty. Yes, it is accepted, but at certain ground we have to make specific or standard procedure or the methodology or a specific dose while prescribing the medicine. So when we talk about posology, so still it is a question mark in our homeopathy. We talk about susceptibility, we talk about idiosyncrasy, we talk about the age and the gender criteria. Whereas in modern pharmacology, they have a specific answer, like 0.5 ml of oil is going to apply on the alopecia model in the rats. So they have a specific dose, 0.5 ml. What about in homeopathy? So that's why we are going to make this specificity, or what we can say a specific order in the dosage or in the action of the homeopathic molecule, so that it can be accepted as a evidence-based medicine.
thank you very much, and congratulations for your studies and research. Are really amazing. Thank you so much. Thanks.
Thank you
Dr.
so much, ma'am. Thank you so much.
Thank you, Dr. Maria. And I think Dr. Ram Parvesh sir asked one question here. You can directly ask. We have 7 more minutes.
Homeopathic Q power, drugs power. What is drugs power?
Yes, sir. Drug power, sir. Basically, this drug power denotes the basic, what we can say, the drug vehicle composition, in how many concentration this drug molecule is going to propagate or divide it or fractionate in the vehicle proportion. So this will denote you the drug power. Suppose if it is 30C, so the drug, basically the drug strength is 1 by 10 to the power 30. So it is 30 times it is fractionate in the vehicle, and it is 30 times it is dynamized, not a diluted, it is dynamized or fractionate in a vehicle base. So this drug power denotes you the fractions in between the drug and vehicle ratio. If it is more fractionate, if the drug vehicle ratio is more propagate and the fractionate or divided, so the efficacy will be more finest and more dynamic. If the drug vehicle ratio is less fractionated and divided, so the efficacy is less as compared to the more one. So this drug power, it always defined as a, in terms of metric system or the modern pharmacology, we can say this drug power is a fractions or the division in between the drug concentration along with the vehicle concentration. So if it is more fractionated, so the dynamization is more. If it is less fractionated, then dynamization is less, then it is more towards the modern pharmacology. So again, the drug power plays a major role. That is the biggest, what we can say, feature and the specific characterization of homeopathy, because in allopathy they have a concentration, they have more mass, they have more density, their drug is not much more fractionate as compared to the vehicle proportion, whereas in our homeopathy the drug is more fractionated as compared to the vehicle in this particular system. So that's why it is a beauty, and that's why it is called dynamization and a potentization.
Yes, yes, thank you. Every Q potency difference drugs power.
Sir, basically every Q potency may not have different drug power, because Q, it denotes the mother tincture. So what is mother tincture? Because mother tincture, it is a crude part. So when we took the extract or any raw material like dried or fresh leafy vegetables, non-leafy vegetables, so when we have this concentrated part in a hydroalcoholic base as per the pharmacopoeia, so they have a similar, not same, they have a similar drug strength, but it may vary from different sources, it may vary from different alkaloidal composition, it may vary from different what we can say phytoconstituents, primary metabolites and the secondary metabolites. But when we talk about the mathematical expression of this mother tincture, it is always similar to all, either it is from plant, some other vegetable group, they are similar. But for the alkaloidal nature, the chemical property of this particular view, then we can say yes, it may vary at certain time. That's why different drug having different action in mother tincture also.
Thank you, very thanks. अरे
Yes,
मनोज
sir.
कुमार का गाना है हिंदी में कि होम्योपैथिक के लिए पानी रे पानी ते रंग कैसा, इसमें दो लगे उस जैसा. It is नैनो टेक्नोलॉजी.
Correct, sir.
Thank you.
Thank you so much.
Thank you so much. And now for giving, Dr. Danesh came. Dr. Danesh, please.
Congratulations, Dr. Suraj Singh Bhadoria. It's really a very good session. And we used to say that the mother tincture is considered to be the 1x potency.
Yes, sir. Yes, sir.
There is a confusion among people that both are different, but in our textbook it is written that the mother tincture itself is the 1x.
Yes, sir.
Okay, now, Doctor, have you finished? Okay, I'm inviting Dr. Mariam Salcedo for giving vote of thanks to Dr. Suraj Singh Bhadoria. Please.
Thanks so much, Dr. Rajini ji. It's a pleasure to be here sharing with you all. And thanks so much, Dr. Suraj Singh Bhadoria, because you did an amazing conference, an amazing research, and thanks for bringing us the evidence for our important medicine as is homeopathy, so we can spread it with a scientific basis. So thank you very much to be working in this path. So we are waiting for you, for your new research, for your new hypothesis, just to follow up your steps, and then we can learn and grow together. So thank you very much, and welcome always to this IFPH family. Thank you all for joining us.
Thank you so much, ma'am. Thank you so much.
So now it is the time to move to Malayalam
Thank you.
session. Thank you, Dr. Suraj Singh. Thank you,
Thank you.
Dr. Mariam Salcedo, and those who are joined for today's session. Actually, IFPH is growing every day. We have researchers with us. We have many good faculties with us. So our journey is continuing, and we are continuing our success day by day. Really thankful to everyone, those who are joining with us and encouraging us, and the participants. Love you all, and we are moving to Malayalam session. I am inviting Dr. P. Shaila for moderating Malayalam session. Bye bye. Good night to everyone. Thank you.
Thank you, ma'am. Thank you.
वाह वाह बहुत अच्छा